The second unique property that I want to mention is that LTP is input specific. And I'm going to come back to this figure to show you exactly what I mean here. So basically what I mean by this is that when induced at one synapse, we don't see the other synapse demonstrate LTP. No synapse. Oops. No LTP. So I go back to this circuitry here to show you what I mean. Right? Remember, they used a tetanus. They used high frequency stimulation to show that, right? So before tetanus, we saw this EPSP, and then after the tetanus, we saw this EPSP, and we described that as LTP. These neurons, as I mentioned before, target the same CA1 axon, or CA1 dendrite, excuse me, right? So here's the CA1 dendrite. And so the question is, does inducing LTP in one neuron induce it in the other? And as you might expect, that answer is no. And so I didn't point this out last time, but see what these labels say? This is before tetanus to pathway 1 and after the pathway, or after the tetanus to pathway 1. So they're still going to, right, if this is our pathway 1 here, they're still going to tetanize this and measure pathway 2, I'll do this in a different color, to see if they can see any LTP occurring here. And the answer is no. So before they've done anything to pathway 1, right, they get this response, and then they tetanize pathway 1, right? So here's the high frequency stimulation. And we know what this does the next time they stimulate this pathway, right? They see LTP. Here's the stimulus. But does this change what's going on in pathway 2? And the answer is it doesn't, right? So after the tetanus to pathway 1, that response is still the same. So even though LTP has been induced, and we can measure that in the postsynaptic cell after, we cannot measure it in the second pathway, okay? So the idea is that this is specific. If we want to measure LTP here, we have to stimulate here to get that measurement. Stimulating here and trying to record LTP doesn't do anything. Talk about the third sort of feature, unique feature of LTP. And that would be associativity. Right, which is basically, we can define this as the selective enhancement of jointly connected synaptic inputs. Okay, so let's look at this circuit down below. Panel A, right, really shows more of what we just looked at last time, input specificity. I think I crossed my F. The idea here is that if we provide high-frequency stimulation to pathway 1, this causes us to see LTP when pathway 1 is stimulated after that high-frequency stimulation. And so that's, I think, what we would expect to see if we give HFC, high-frequency stimulus, and then wait for a little bit and stimulate again, we see facilitation. But that LTP is independent of whatever happens in pathway 2. Pathway 2 is not strengthened. Right? So even after we induce LTP here, right, it's the same neuron we're measuring from, right? We're measuring from this one. Any stimulus to this neuron won't show LTP, right? Input specificity. But we wanted to talk about associativity. So let's look at pathway B here. Well, we know that pathway 1, just like we said last time, high frequency stimulation leads to LTP when we stimulate after the high frequency stimulus. Okay. But pathway 2 does not induce LTP. When the presynaptic neuron is providing only weak stimulation. All right, so what we're saying here is if this is some amount of time, if there's only one impulse or two impulses that reach this terminal, that's not enough to give us LTP. I don't think that's a surprise. What may be surprising is that when these are happening at the same time, so we'll say, but when high frequency stimulation in pathway 1 is paired with even weak stimuli, of pathway 2, both postsynaptic responses show LTP. Right, so it's the idea that if we're giving high-frequency stimulation here, and this is occurring at the same time, right? So the temporal effects matter here. So if this stimulus occurs when this high-frequency stimulation is occurring, this synapse will be strengthened. Okay, time to talk about molecular mechanisms that are underlying LTP. These are all those pathway things that we've just talked about. Okay, so initially we knew that ionotropic NMDA receptors were involved. So I can tell you how in a second. Right, so hopefully you remember for NMDA receptors, these are ionotropic receptors, right, that open in response to glutamate. So the ligand is glutamate, and they are ionotropic, ion channels, right? And we knew this because antagonists had been developed that block glutamate receptors, okay? So we had antagonists. All right, so these are going to inhibit the channel. Agonists are things that activate the channel. Ligands are agonists. Antagonists are things that block the channel and thereby their effects, right? So antagonists block NMDA receptor effects. And so when we added antagonists into the bath where the experiments were taking place, we eliminated LTP. But we still saw a response when the cells were stimulated with a lower frequency. Under low frequency. Okay, so if you think back to the experiment that we just did, right? If we were to try to induce LTP by giving a train of action potentials, and then we gave one stimulus, we wouldn't see any different from the control, right? We'd still see the control, right? That's what this would be. This response was still seen, but the LTP wasn't. And that effect was only seen in the presence of antagonists. So we think that antagonists of NMDA receptors were doing something, which leads us to believe that NMDA receptors were involved. All right. I think before we talk about exactly what happens, we have to talk a little bit more about these NMDA receptors. So recall NMDA receptor properties. Remember, these receptors were really unique, right? Yes, they were ionotropic, but they also required a co-ligand, glycine. Right, so both have to be present, and they require depolarizing pulses. So there had to be a requirement of something to alleviate what was magnesium blockage, remember? So required magnesium that was present in the pore of the channel to be extruded. And the way that magnesium was extruded, right, was some sort of depolarization. So the NMDA receptor wouldn't pass current unless glycine was present and the cell was already depolarized, because that gets rid of the magnesium. Okay? The last thing that I'll tell you here before we take a break and go on to the next video is that glutamate could bind to another type of receptor, if you recall, right? So somewhere up here, maybe down here, I'll put that AMPA receptors also bind glutamate. They're also ionotropic. And they don't have any of those other odd properties. They don't have magnesium that blocks the pore. They don't require a co-ligand like glycine. AMPA receptors, I think, are a little bit more traditional in that when glutamate binds, the AMPA receptor opens up, okay, and it passes cations through it. So we'll come back and talk exactly about how molecularly LTP can be induced.